bacterium present in many foods, and when not cooked well or prepared properly, it can cause food poisoning to its consumer. Bodies of water, which may be contaminated with germs from animal or human waste, may also cause a variety of symptoms, such as gastrointestinal, skin, ear, respiratory, eye, and wound infections.
Welcome, guys and girls, Matrix of South Africa. My name is Kathy Hasty, and it's Wednesday, so of course we're doing life sciences. Before we start though, a huge thank you and shout out to Liberty. They are in it with you so that you can achieve the best prelim results possible. Blah, that's like an alliteration right there. So, guys and girls, don't forget to download your Tenfold Education app. It's got a tutor function, so you can submit questions. It's also got a process where you can live stream our lessons if you don't have access to a television. Then we've got our WhatsApp bot text, so Dex, so you can send your questions and through to Dex. Please make sure that you take a good photo of the question, or else we can't use it, all right? And then don't forget the YouTube channel at Mindset Learn, where you can watch all the videos that we do here. So there is no excuse for you not to have every kind and form of assistance possible to get the best results and absolutely ace your exams, all right? So here we go. Oh, my quote of the day, I keep forgetting, and thank heavens for the wonderful tech staff that we have here. My quote for the day to you is, some people dream of success while other people get up every morning and make it happen. So you can't make it happen if you just dream about it. You have to get up, have that coffee, and get ready to slay that dragon. And this is what we're going to do today. So our first question, glasses. This first question comes from Sinalo. Let me just quickly zip this up. This is from Sinalo. So thanks for sending this in. It's a very, very nice question. Okay, and it shows a visual defect. Now remember, people, the questions we do here are not going to be in the prelim exams or in the final exams. These are all questions that have either been in tests or previous examinations that learners are sending in because they're using them to revise. What I do, or I try to do when I go through these questions, is explain how processes occur to help you understand the structure and how it functions, so that if they ask you any question on the structure, you'll get full marks. That is the aim of this. So we just use the questions as a sort of a jumping board, a starting board. All right, so here we go. What do we see, first of all, when you look here? The first thing is A. What is A? That is the, come on, you've got to know, the cornea. And you'll see the cornea has a nice rounded shape. So it is convex shape. It bends outwards. And therefore, it refracts light. Okay, then we have B, which is the second layer here. And B is the choroid. And the choroid layer contains the blood vessels, which carry clearly oxygen and nutrients. And it also has a pigment in it, which is going to absorb excess light. Then we look at C, this little round number here, and that is your lens. And the lens is important because it is the lens shape that changes for accommodation to see far and close. This point here is called the yellow spot. And the yellow spot, the center of the yellow spot is called the fovea centralis. Now, the fovea centralis only has cone cells, but the yellow spot has a huge concentration of rod and cone cells, okay? Why? Because this is where the image is formed that gets sent to your brain, to the cerebrum. All right, now, this is the optic nerve. This point here for E is called the blind spot. And why? Because the blind spot has no rod and cone cells. Because would you put furniture in front of a doorway? No. Okay? And that optic nerve, where it exits the eye, it's like a doorway. It's the doorway to the impulses. So there are no rod and cone cells at the blind spot. Then F is the sclera. And the sclera is white and fibrous, and it's there to protect the eye and maintain shape, as well as the vitreous humor that's in here and the aqueous humor that's in the front of the eye. And then G is clearly the iris. And remember, the iris extends from the colored choroid layer as well. So you've got pigment in the choroid, and it runs all the way through into the iris. Okay, and then the hole between the iris or inside the iris is the pupil, which is in the center. Now, what you've got here is an image that is going to fall short of the retina. Therefore, this person would be short-sighted. Now, you see all the stuff that's given to you here in one diagram. Okay, so this person is short-sighted, and to correct this image that is falling short of the retina, because if it doesn't fall here on the retina, well, the photoreceptors, the rod and cone cells, won't be able to convert the stimulus to an impulse for it to go to the cerebrum to interpret light, images or what you're seeing. So how would you fix this? Well, in order to fix short-sightedness, you would need a bi, means two, a bi, and look here, they concave. So it would be a biconcave lens. Okay? A concave on that side and a concave on that side. Bi means two, concave lens. And what that does is it diverges, which means it makes the angle here so that this rounded shape of the eye, it undoes that basically by making sure that it diverges light rays before they enter the eye. And then they go through the cornea and they go through the lens, but because they have been diverged, they will then be able to, let me do it in blue, they'll be able to extend and fall onto the back of the retina. That's what a divergent lens does. If, for example, the image, and they show you the image falling behind the eye over here, it means the person is farsighted. So what would happen then, they would then use a biconcave lens. This is, I'm going to put you far-sighted. So that image falls after the retina. So they would use a biconcave lens, and that biconcave lens would look like this. So concave, what, not concave, I'm being such a doofy here. Just hold on, let me just zip, zip, zip. Sorry, a biconvex lens, because there's the convex shape, there's a convex shape. So it's a bi to convex lens, and it is not going to diverge, it is going to converge the image or the light rays, and that means that the light rays are going to be sorted out and converged before they hit the cornea and the lens, and they are going to then be shortened so that they fall onto the retina. Okay, so if they can give you the same diagram and ask you about the functioning of the iris, they can ask you about the functioning of the cornea and the shape of the cornea, and the functioning of the lens, and the functions of the choroid, and the yellow spot, and the optic nerve, and the blind spot. Okay, so a lot of things on the eye can be asked just from this diagram. So in an exam, you're not going to get the same question for your prelims or your finals, but you may get a biconvex lens, okay, and not a biconcave lens. All right, so let's look at our questions. Identify F, well, that's easy, it's the sclera. Okay, then state one function of B. What was B? B, 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 B is the choroid. Okay, so that's the choroid. So what does the choroid do? It has blood vessels to supply eye cells with oxygen and nutrients. And since this is a science, you can write O2 instead of writing oxygen out. Okay, the second thing, remember I told you, is the choroid contains a pigment. And what does the pigment do? The pigment absorbs, contains a pigment, oh, it should be that absorbs, no, it should have an R there. Absorbs excess light, therefore prevents reflection or internal reflection. You don't want light bouncing around inside the eye. So any excess light is absorbed or is absorbed by that pigment. Okay, that's also when you take photos of of people with a flash, their eyes look red. Well, that is the pigment that you're actually seeing. Okay, so here we go. Explain why the light rays must focus on D rather than E. So D is the yellow spot, and E is the blind spot. Now, look at this question. It says explain. And when they say explain, they want you to be able to tell them what each structure does. So we start like this. We'll say part D. Okay, there's the yellow spot. Has the highest concentration of photoreceptors. Remember, it is only the fovea centralis that has only cone cells, but you have the highest concentration of photoreceptors, so rod and cone cells, at the yellow spot. Okay, so it has the highest concentration of photoreceptors, so therefore the clearest image will form when light falls on it. Okay, now let's just make this come down a bit. And then we look at, it was part E. And part E was the blind spot. This is easy. The blind spot has no photoreceptors. So receptors. Okay, so logically, therefore, no image will form when light falls on it. I mean, that's logic. The yellow spot's got all the photoreceptors, so when light falls on it, it has the clearest image of the yellow spot. And the blind spot, well, there are no photoreceptors, therefore there's going to be no image. You need photoreceptors to take that light stimulus and convert it to an impulse. Okay, now, name the type of lens that is used to correct the visual defect shown in the diagram. As I said to you, this is short-sightedness. And if it's short-sightedness, we are going to use a biconcave lens so that we can diverge the light rays before they get into the eye. So, name the type of lens. It's going to be a biconcave lens. Okay, or if it's both eyes, biconcave lenses. And I'm going to just put in brackets here so that you remember. Biconcave. So there's a concave and there's a concave, and the two together, biconcave. Bi means two. All right, and then describe how the type of lens named in that one, so biconcave lens, corrects the visual defect. Okay, well, that's pretty easy. Biconcave lenses diverge light rays before, and this is important, before they're going to hit the cornea or the lens, so they diverge light rays before entering the eye. Therefore, refraction is reduced because it's got an image that, or it's got a light rays that are now less bent, so it reduces. Where am I now? Biconcave lenses diverge light rays before entering the eye, and therefore refraction is reduced to allow light to focus on the retina, and it's actually the yellow spot. So I'm going to just put that in brackets here for you, but it's the retina, okay, to form a clear image. And then that image is what the photoreceptors will convert into an impulse and zip, zip, zip out the out the blind spot, down the optic nerve, to the brain, to the cerebrum, actually to the occipital lobe in the cerebrum, where what you see, that image, is going to be interpreted. It's turned upright and it is interpreted. Okay, then, where are we here? Explain one way in which part C is structurally suited for accommodation. Now, the only thing that's structurally suited for accommodation that we've got see anything here is the lens. So how is the lens structurally suited? Well, first thing, the lens is elastic and is able to change shape to focus light rays where onto the retina. So what actually changes the shape of the lens? The lens itself can't change its shape. The lens is the lens. It sits here. There's your lens. It doesn't change shape. What changes the shape of the lens are these suspensory ligaments and the ciliary muscles. So when the ciliary muscles contract, they pull towards the lens. The suspensory ligaments become slack. The lens becomes rounder. And when the lens is rounder, okay, it is then going to refract the light more, which means we'll be able to see things that are within or closer than 6 meters. Okay, that's if it's rounder. When the lens is flatter and longer, how does that happen? Well, the ciliary muscle says, you know what, I'm going to relax, and it pulls back. So people think about it. When you want to relax, do you sit like this? No, you chill out. You lie back and you chill out. The same with the ciliary muscles. They say, oh, I'm going to chill out. And they relax and they pull back. And when they pull back, well, what's attached to them? The suspensory ligaments. So those suspensory ligaments pull tight. So they become taut. And as they pull tight, they pull this lens longer and flatter. So that the lens becomes longer and flatter and less refraction of light. Okay? And that would be if it is further than 6 meters. That's what's going to happen. All right, you need that lens to be long and flat because you don't want the light bent. You want the light to just go through normally. Okay, was there any other questions here? Let's just check. Oh, name 2 muscles that form part of G. I'm assuming G was the iris, correct? So that's the iris. So the 2 muscles are the circular muscles and the radial muscles. And remember, these 2 muscles work antagonistically. So when one contracts, the other relaxes. When this one relaxes, this one contracts. And all you have to remember is this. You see my hand? Okay, you see the C shape? Okay, when the circular muscles contract, the pupil constricts, it gets smaller, and the diameter, as the diameter decreases, okay, or the diameter decreases as the pupil constricts to allow less light into the eye. So when would that be? Well, when it's very bright. And the opposite happens when you're in a dark place. So in dim light, the circular muscles relax. So therefore, the radial muscles must contract to open it up. So the diameter of the pupil increases as the pupil dilates, allowing more light into the eye. So you only have to learn one, and you've got the other one. That's called the pupillary mechanism, or a pupillary function. And then name and describe the visual defect that occurs when part A is uneven. Let's just check. I think A was the choroid. Yep. I mean, the cornea. When the cornea is, so when it's got a little ding there, or it's got a little blip there, or another little ding there. Okay, so it's uneven. So space, space, space. Let's go up. So name the visual defect. It's called astigmatism. Okay, astigmatism. Okay, and how the visual defect occurs when part A is uneven. So that's pretty easy. Light is refracted, which means the light will be bent. So instead of a nice smooth surface, it's now got a surface that looks like this. Okay, so obviously light is refracted because of the shape of the cornea. Unevenly, so it's not even like it's supposed to be, and becomes distorted, all right, forming a blurred image. Where? On the retina. Done and dusted. So it's not smooth, it's going to be uneven. Okay, guys and girls, are there any other questions here? No, that's it for this one. I think it's time to take an ad break. So let's go have our ad break, and I'll see you all back here in 2.3 minutes.
No one ever asks why we do the weird things we do. Like, why once is never enough. Or doing 2 trips is just one too many. Why do we, before we use these, or say 5 when we mean 20? Why do we have second thoughts? And think that lift buttons make them go faster? Why don't we walk there? Cross our fingers, touch wood, and struggle to hide our true feelings. At Liberty, we want to know all the weird and wonderful things that make us human, because human beings, being human, are what we make financial solutions for. Liberty, in it with you.
Want to catch up on some of your studies? Join Mindset for late night studies. Monday is mathematics, Tuesday is physical sciences, with life sciences on Wednesday and accounting on Thursday. Friday is English, maths literacy on Saturday, and Sunday's a good day to catch up on geography. Every day from 9 p.m. till midnight, and again from midnight till 6 a.m. Late night studies, only on Mindset.
And I'm live. Welcome back, guys. Before we start, huge big thank you to Liberty. They're in it with you. You are going to rock for those prelim exams because they are assisting you in every possible way. So there you go. Guys and girls, don't forget to download your Tenfold Education app. It's got a tutor function. It's got a whole bunch of stuff on math, science, and life sciences to help you. And you can live stream. And then don't forget our WhatsApp number, our little bot called Dex. He's actually very cute. And he scuttles around here looking for questions all the time. So just remember to send in your life science questions next week. Next week, we are doing paper 2. So we're doing prep for paper 2 because you are writing paper 1 on the 3rd of September. Okay, so I want you to rock that exam on Friday. But for the next one, send in those questions. Start sending them in. Becky collects them for me, and we will make sure that the first 5 or 6 are ready to rock and roll while we go through our lesson. And then don't forget our YouTube channel, at Mindset Learn, and you are able to look for any videos on the different sections that you want to and topics you need to cover. Okay, so let's start with our next question. Okay. This one was sent in by Karabo. And Karabo, thank you, hun, for sending your quizzy in. And let's go with orange. All right, it says, I hate these read the extracts. Okay, so, this is reproduction in guppy fish. So now we're looking at reproduction in vertebrates, actually. So just before we carry on, I want you to know this. For reproduction in vertebrates, okay, you have to know oviparous or ovipary, viviparous, viviparous, and ovoviviparous. Okay, so oviparous, remember the O, they lay eggs. Viviparous, think of the IV for live, okay? And the O equals eggs for oviparous. And ovoviviparous, well, it is eggs plus live. So the eggs are kept inside the female, and they hatch inside her, and then she gives birth to live young. That is how you remember it. Something else you must remember is internal and external. For heaven's sake, internal fertilization happens inside the female's body. External fertilization happens outside the female's body. I mean, really, that you learnt in grade 4, actually. And then you must know altricial development and altricial and precocial. I had to think there. Development. Now, with precocial development, a child, when people say that child is precocious, it means that child is grown up for their years, or they think they're grown up for their years, and you say that kid is precocious. That's where the word precocious comes from. That's why they talk about precocial development. It is the babies are born, they hatch or they're born, and they can pretty much take care of themselves. They can fend for themselves, they can feed themselves, they can run away from enemies. All right. They're protected by their parents, sure. I mean, like when a foal or a buck gives birth to a baby buck, that baby buck, clearly the mother will look after it and she'll nurse it, but it will also be able to graze. So, and it can run very fast. So that's precocial development. They can take care of themselves. Their eyes are open when they're born. They can move immediately or pretty soon after birth or hatching, etc. Altricial, think about altricial as the parents must do all for those children. Okay, they must do everything. Those babies are born and they are useless. They cannot feed themselves. They depend on their parents for food. They depend on their parents for warmth because they can't control their body temperature. They have no body covering. They're blind. They can't move. Okay, so they are completely 100% reliant on the parents. Altricial, the parents must do all to keep them alive. Precocial, they're precocious little brats. They're ready to rock and roll from the time they're born. That's how you remember it. Those are the most important things, and they're really the only things that they can ask you about in the exam are these 5 things. Altricial, precocial, oviparous, viviparous, and ovoviviparous. Combination of the two. So here we go. Reproduction in the guppy fish is very interesting method of breeding. During the mating, okay, listen carefully. During the mating, the male deposits pockets of sperm inside the female's body, and it should be female with an E, reproductive opening, using an organ called a gonopodium. Okay, so I suppose that's like a little mini penis type structure. This process takes place several times, and the female stores some extra sperm. Yay. All right, now, the fertilized eggs remain where? They remain inside the female's body. But it tells you that they are eggs, so there is no placental connection. Means there's no placenta. And no placental connection is what we have when we have viviparous development. When it is viviparous development and the female gives birth to live young, there is a placental connection. The baby is connected to the mother via placenta. So here we've got eggs which stay inside the female's body until they hatch. And then the young are born live. So now we know this is going to be ovovivipary. Okay, right now. We got the male fish, we got the female fish, which was actually quite unnecessary, but anyway. Name the type of fertilization in guppy fish. Well, the male inserts the sperm inside the female, so it must be internal fertilization. Sation is American, zation is English. You use whichever, they're both correct. Okay, I'm old, so I like z. And my computer is set to Z, so Oxford English. Explain two ways in which the types of fertilization named, it says which the type, it should be S's out, of fertilization named in question 214 increases reproduction success. What is reproduction success, people? It means that as many of those ova are fertilized, and that as many of them make it to adulthood. That is reproductive success. So think about it. Use your common sense. So the sperm is deposited inside, and it tells you, inside the female body. Therefore, increases chances of fertilization. Okay, they're not being sprayed over in the water somewhere. Okay, so the sperm is deposited inside the female and it increases those chances of fertilization because the sperm is right where the little egg cells are. And the embryos are protected by the female body against predators and the environment. Remember, where do fish live? They live in water. What happens in water? You've got currents, you've got storms that blow around, and these poor little eggs move all over the show. Okay, in some cases it's good because it distributes those little embryos. But here, the embryos are inside and they're protected in the mama's body. All right, the mama fish, the female's body. Why are guppies regarded as ovoviviparous? Oh, please, people. I mean, that is the easiest 2 marks you're ever going to get. The eggs, and it tells you that in the actual question. The eggs hatch inside the female body and the young are born live, or you can say she gives birth to live young. That's it, 2 marks. I mean, jeez, 8 marks in the bag. Bring on the rest of the 42 marks that you have to get for the exams. Okay, how easy was that? All right, now, where am I going? This way. Okay, reproduction in, here we go. Human reproduction. I've never understood why you guys battle so with human reproduction. It is a piece of cake. So, what have we got? Thank you, Junior, for sending this in. You are an absolute star. And now, let's look at our question. The diagram below shows the ovaries and the uterus, okay, are changed by various hormones during the menstrual cycle. The diagram below is not drawn to scale. Well, I would hope it's not. Okay, because if that's how big everything was, we would be in trouble. Okay, so here we go. This is day 1 to 28. And here we have the lining of the uterus. What's that called? It is the endometrium. The uterus has got 3 layers. It's got the epimetrium, which is on the outside, then the myometrium, which is in the middle, it's the muscle. That's what contracts to push those bubbas out. And the endometrium is the inside layer. Okay, so there's our pituitary gland, and it tells you that this little very strange, I just want to have a sip of this water. Okay, you've got the pituitary gland, which is drawn very precariously, looking like testes, but we won't go there. Okay, now before we carry on, I just want to show you this. Your pituitary gland, okay, secretes what hormones? They are all the abbreviated ones. You've got growth hormone, follicle stimulating hormone, luteinizing hormone, antidiuretic hormone, thyroid stimulating hormone. The only hormone the pituitary gland releases that can't be abbreviated is prolactin, which is released when a woman gives birth so that her mammary glands will produce milk. Okay, but all the others, if it can be abbreviated, it is a pituitary gland hormone. Easy peasy. Okay, so now, your ovaries, okay, so what happens? The pituitary gland releases this hormone here. And this hormone here is going to be follicle stimulating hormone, which does exactly that. It stimulates the follicle to develop, okay, and this little guy here is called the Graafian follicle. Okay, so this little follicle develops and it grows. But as the follicle develops inside the ovary, it releases estrogen. Okay, so think of ovary O, and estrogen starts with an O. So it releases estrogen, and the estrogen causes the endometrium to thicken, and it becomes more glandular and vascular, which means it's got all vascular, so that it has all these blood vessels, and it's glandular. Why? For implantation. So that there's a nice mushy place for the embryo to implant when fertilization takes place. Okay, now the Graafian follicle releases this little guy, which is an ovum, and the ovum is released during ovulation. Ovulation is the release of the ovum by the Graafian follicle. Another definition. Now, the pituitary gland, when there's lots of estrogen. So first, the pituitary releases follicle stimulating hormone. That stimulates the development of the follicle, and the follicles release estrogen. And what does estrogen do? It makes the uterus lining increase and thicken. Okay, now lots of estrogen stimulates this strange looking pituitary gland here to release this hormone, luteinizing hormone. So we go follicle stimulating hormone, estrogen. Lots of estrogen causes the pituitary, so that's the pituitary, this is the ovary, to release luteinizing hormone, okay, which is from the pituitary. And luteinizing hormone converts, first of all, it causes the Graafian follicle to release the ovum, so it causes ovulation. And what does luteinizing hormone do? It causes this Graafian follicle to develop into this one, which is the corpus luteum. And what does the corpus luteum do? Very special, tiny little structure inside the ovary, releases progesterone. So we go follicle stimulating hormone, estrogen, luteinizing hormone, progesterone. So remember FOLP, and this is POPO, popo. So pituitary, ovary, pituitary, ovary. Okay, easy peasy. I mean, How difficult is that? If you remember what I've written on here now, I promise you now, they cannot give you a question on the menstrual cycle that you will not get full marks for. So let's go through this quick. Name hormone V. Hormone V is estrogen. Okay, describe the effect of estrogen on the lining of the endometrium. Well, it causes the endometrium to thicken. Or you could say it causes the endometrium to become more glandular and vascular. Okay, explain the importance of the endometrium lining. Well, guess what? And it's for 2 marks, so you've got to give 2 importances. Okay, so the first one is that it provides a surface for implantation of the embryo. It's actually the blastocyst, because it implants at the blastocyst stage with the chorionic villi that grow into the endometrium surface. So, but if you just wrote embryo, it's cool. You get your mark. And then, for the development of the placenta. And that's incredibly important, because if that placenta doesn't develop, that embryo just gets pushed out and a miscarriage takes place. Okay, so with reference to the diagram, name hormone X. So let's just quickly check. I think hormone X was it's luteinizing hormone. Okay. Okay, so it is LH. Just write LH so that you don't make spelling errors. Okay, so LH. Give the functions of hormone X in the menstrual cycle. Well, as I showed you here, the first thing, luteinizing hormone causes the Graafian follicle to release the ovum during ovulation. And what does it do? It causes the Graafian follicle to develop into the corpus luteum. So that's all you're going to write there. Should I write it? Okay, let's write it. It stimulates ovulation. Okay, and it stimulates the development of the corpus luteum. People, you must know how to spell corpus luteum. You must know how to spell ovulation, okay? Then, give 2 visible reasons from the diagram that indicate fertilization did not take place during this menstrual cycle. Let's have a look. Okay, let me get another color. Okay, first thing is, can you see that the corpus luteum starts to disintegrate and get smaller here? So it gets smaller because it's starting to disintegrate. Why is it disintegrating? Because fertilization didn't take place. We no longer need progesterone. Secondly, look here. Can you see? This is menstruation. And then the estrogen increases. And as the estrogen increases, so the thickness of the endometrium increases. But look what's happening here at day 28. The endometrium lining starts to decrease. So it decreases in size. So number 1, the corpus luteum decreases in size and starts degenerating. Number 2, the endometrium does the same. So how are we going for time? Yeah. Okay, so 2 visible reasons. The first visible reason, the corpus luteum, you can either say starts to degenerate, or you can say it starts to decrease in size, which means it is degenerating. And then number 2, you say the endometrium you don't have to say endometrium lining. The endometrium is the lining of the inner lining of the uterus. That's what it is. So you don't have to say the endometrium lining. So it's just the endometrium is decreasing in size towards day 28. Done and dusted. And that is the last question. Okay, we are scrambling for time here. So I've got another question. Let's try and get through this one. Okay. Oh, this is from Nongamzo. Thank you, Han. Here we go. Question. The diagram below shows the regions A, B, C, or A, B, and E. So there's A, there's E, and there's B. Okay. Of the nervous, without an E. Okay. Of the nervous, without an E, system that can be blocked by a local anesthetic. People, a local anesthetic is the thing that, when you go to the dentist, all of us have gone to the dentist at some other stage. What do? They give you an injection so they can drill in or take a tooth out. That is local anesthetic, okay? So it's a drug or agent that produces a complete or partial loss of feeling for the various medical procedures. So here we've got the brain, we've got the spinal cord, the two form part of your central nervous system. We've got A is the first blocker. They tell you here, block. The A is the block at A. B is a block at B. So this is your thumb. And in your thumb, you're going to have receptors. And in the muscles in your arm, you're going to have effectors. Okay. There's your impulse going this way. So C is your sensory neuron. And D would be your motor neuron. Okay. And there's your impulse comes back here, and that will cause the movement. All right. Remember, it always goes receptor. And the receptor converts the stimulus to an impulse. And the impulse travels along the sensory neurons to the central nervous system. So in a reflex action, okay, you are going to, it'll only pass, or a reflex arc at least, it will only pass through the spinal cord. And if it goes all the way to the brain, then it's still the central nervous system. Okay, so sensory neuron to the central nervous system, and from the central nervous system it goes motor neuron, and from the motor neuron it's going to go to an effector, and that could be a gland, it could be a muscle, and it will cause a response. Okay, if it only passes to the spinal cord, it is a reflex arc. So your reflex arc would be your sensory, your receptor, which is in the thumb, and it's going to go sensory neuron, and then it gets to the spinal cord, and over here we'll have a connector neuron, and then it will cruise and back again along the motor neuron. Okay, that would be a reflex arc. And what do you get from a reflex arc? A reflex action. Okay, and that action would be when the effector moves away or pulls away, etc. Right, so let's have a look at our questions. Write down the correct letter for each statement using the key given. Okay, so we've either got to use A or B or C or F. E or F. So here we go. When the skin of the thumb is stimulated, okay, so we prickle it. Look here. So we take the thumb and we go tick, 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 tick. All right? And the hand moves involuntarily. So there's no voluntary thing. It's involuntarily. But the patient cannot move the hand voluntarily or feel it move. Where do you think the block's going to be? Okay, so look, it moves involuntarily, so it means it's not B or E, but they can't feel it. It means that the block is at A. Why? Because it then becomes a reflex action. All right, then the patient can, whoever typed this up, feel, the patient can feel the thumb being touched or pinched, but cannot move. So look at this now. If they can feel it, what was that now? Sorry. That was they can feel it being touched, but they can't move it. So they can feel it. So B is working, A is working, E isn't working. Okay. The patient can move the thumb, but cannot feel it. Okay. They can move it, but they can't feel it. So they can move it. Moving is working, but they can't feel it. This means this is moving. So what is it? It is going to be B. And if we go here, when the skin of the thumb is stimulated, the thumb moves. And the patient knows it moving, well, then none of them are blocked. Okay, so it would be F. They can feel it, they can sense everything, and everything works. There we go, it's F. Which labeled part of the central nervous system will actively evolve to generate a quick response in a normal, healthy person? Well, it's the spinal cord. Remember, it's an automatic, quick response. Name the type of reaction mentioned in one point. It would be a reflex. It's going to give a quick response. It's a reflex action. And the two parts shown in the diagram, which is protected by meninges, well, I don't even have to look. It's going to be the spinal cord and the brain. Because your peripheral nervous system and the sensory neurons, well, they've got myelin sheath to protect them. Then identify the letter representing the peripheral nervous system, the sensory neurons. What number were they? Sensory neurons are C and the motor neurons are D. So C and D, and we're done. Okay, people, listen carefully now. The format has changed. Number one, there are no essays in your papers. So thank you for that. You've got reproduction in vertebrates is going to count 8 marks. Human reproduction is 41 out of 150. Please make sure you learn it. Okay. Pituitary gland secretes follicle-stimulating hormone. The follicles release estrogen in the ovaries. The estrogen increase causes the pituitary to release luteinizing hormone, and the luteinizing hormone then causes ovulation and the formation of the corpus luteum, which then releases progesterone. And the placenta takes over progesterone production at about 12 weeks. Okay, make sure you learn the reproductive system. Labels, learn your labels. The male reproductive system, know those labels. Female, don't worry so much about the female structure. Make sure you know the menstrual cycle. Reproduction, responding to the environment, the brain, the reflex arc, the spine. Know the brain, structure, function. Know the reflex arc and the reflex action that results. Know the eye, structure and pupillary mechanism, accommodation, and then the disorders. The ear, you must know hearing, the path of sound, and balance, how balance happens. That's it. Responding to environment, plants, that will be one of those, the investigators decided to blah, blah, blah. Remember, your independent variable is the one that's mentioned first. It's the one that's manipulated by the investigator, the scientist. It's what they are looking at, what they want to investigate. And the result is your dependent variable. Okay, so the growth of the stem, the growth of the roots. Know what phototropism, movement towards light. Geotropism, movement towards gravity. Stems are negatively geotrophic but positively phototrophic. Roots are negatively phototrophic because they grow away from the light, but positively geotrophic. Endocrine system and homeostasis. Know your negative feedback system between the thyroid and the pituitary gland. Know your negative feedback system for glucose, okay, to maintain the glucose levels in your body. Know salt and water concentrations. I think those are probably the most important ones. Okay, so, but just know the endocrine system, what the endocrine system is and which organs secrete what. And the difference between an endocrine gland and an exocrine gland, people, please make sure you know that. So, end of the story. That's all I can help you with. Send those questions for paper 2 for next week. Have a wonderful week. Study hard. Good luck for Friday's exam. Mwah! Stay safe.
What's up, peeps? Welcome to Moby School. We are here to make sure that you, yes, you get all the marks that you deserve on the go. We're here every Mondays to Fridays from 2 to 3 p.m. only on Mindset Learn TV. Now, this is a whole hour worth of revision just for you. And I hope that you've got your notepads and your pens ready and that you and your study group are game for today's revision session. Now, my name is Jo, and just like you, I am also in matric, and I'm here for a whole hour to make sure that I ace my final exams, and I hope that you will be doing the exact same thing. Now, do remember that we love hearing from you guys, so log on to our Facebook page, which is www.facebook.com/Moby School SA. Drop us your comments, like our page, share the page, and even spread the Moby School family even bigger. Now, isn't that cool? Yep. Yes, it is. Now, do remember that you can catch us here for a whole hour of revision session where all of our awesome teachers come right here to help you guys revise. So on that note of the first revision session, let's head straight to our teacher.
All right, so I have talked about this already. Mr. Enfield mentions the name of Hyde. Mr. Utterson has already heard the name of Hyde because Hyde is the beneficiary of the will, meaning he will get the money and the property when Dr. Jekyll dies or disappears. That's the strange clause. And where has he put this will? In the most private part of his safe. So all the time I'm going to ask you to make notes about things that are hidden, things that are locked up. And it's in his safe, which is already a space which is not meant to be broken into. And where's he put it in the safe? The most private part, really hidden even within the safe. And we're told that Mr. Utterson would not write it for Dr. Jekyll. It's a copy. And it provided not only that, in case of the death of Henry Jekyll, doctor, and these are all of his qualifications, all his possessions, everything he owns, were to pass into the hands of his friend and benefactor, Edward Hyde, but that in case of Dr. Jekyll's disappearance or unexplained absence for any period longer than 3 calendar months, Edward Hyde should take over in Dr. Jekyll's house with all his belongings, with everything, without any further delay, immediately, and free from any burden or obligation beyond the payment of a few small sums to the members of the doctor's household. Now, this has really offended Mr. Utterson. It's something he doesn't like to look at. It's an eyesore. Because it offends him as a lawyer, and he's a person who likes reasonable things that he can understand. And he says this is fanciful. It's the imagination run mad. It's out of control. And he really doesn't like the will, because it's this, not only the death of Dr. Jekyll, but the disappearance. How can you say if the person disappears for 3 months, then after 3 months you're just going to step into his life? And he says before this, his unhappiness has been that he never knew anything about Mr. Hyde. But now, by a sudden turn, that's what Enfield has told him, he actually now knows about Hyde. So it was bad enough when he didn't know who Hyde was, but it's worse now because he's met the man and he is detestable, he's hateful. So he says it's like being in a mist. So a mist is when like thick cloud. And he says it's shifting and insubstantial. So think about the cloud, it shifts, it moves all the time, and it's insubstantial, you can't kind of grab hold of it. So he disliked this idea of Mr. Hyde. But now suddenly he's met Mr. Hyde, and he says he looks like a fiend, and a fiend is a devil. So he's even more upset about this will. How can this devil inherit everything from Dr. Jekyll? Here we have Dr. Lanyon. Let's have a look at him. He is strong, healthy,